made a few
You gotta read it.
changes to the slides this morning, and for some reason, when I pulled it up my desktop down here, the new version didn't come up, so anyway, I'll get you the new version. The issue that I mentioned earlier about the being able to smell your sister is right there. One point I want to make about smell, though, is that the uncus, as well as the limbic system, but the uncus in particular, it has the lowest threshold for hypoxia. So during childbirth, during delivery, if there's hypoxia of the brain with that would cause abnormal development of neural tissue, the uncus is going to be infected first. As you get older, if there is aberrant or epilepsy associated with aberrant electrical activity originating in the uncus, because it's the most likely damaged, these people have a prodrome to their seizures. In other words, it's a sensation that they get just before the seizure activity happens. And if it's in the uncus, it's a noxious smell. They smell something that is wholly awful. They don't, this is not a smell they can say it's like rotting flesh or, rotten fruit or something like that. This is a smell that they always describe as just otherworldly bad. It's never good for some reason. It's never good. It's always horrible. And then they go out with a seizure. That's called an uncinate fit. It's an old term, been around since like 1890s. That's when it was first characterized. But people still use it. Uncinate fit, or temporal lobe epilepsy, can start out with a smell hallucination. It's a hallucination. All right. This is under the gustatory thing. PTU, phenylthyrouria, is a compound that has a genetic component to it. It's a chemical that, if you have the protein to act as a receptor for it, you can taste it. Other people who are homozygous recessive, since it's a dominant trait, you have to have two recessive genes. They can't taste it. It's a very strong, for those of us tasters, I can taste it. It's strong. It's like putting battery acid on your tongue. Not painful, but it's like putting, well, I don't know what it is. It's very strong. There's no mistaking it. But other people can just bathe their tongue in it and not taste it because they lack the receptor for it. What's that?
I can't taste it.
You can't taste it? Yeah. And the other thing you have to watch out for in terms of taste and smell is that a lot of taste are aerosolized. A lot of chemicals that have taste are aerosolized. Like if you have a steak, you not only can taste the steak, the salt, the whatever the other tastes are, but because it's vaporized, cooked meat is vaporized, you smell it. They come together to form the flavor. Or if you're a vegetarian, a piece of broccoli. Okay. If you look at the taste buds, this is what a taste bud looks like. You have different types of papillae. you have three different types of papillae, you remember from anatomy. You have the foliate papillae, which are the tiny little folds of tissue. You have the fungiform that look like a little mushrooms. And then you have the vallate papillae that are in the back. The fungiform papillae, they only have like a half dozen taste buds on them. These guys, as well as the vallate papillae, they have a lot of them. And you have to appreciate that this is a taste bud. It's made up of several cells that are tied together through what are called tight junctions at the top. In other words, they don't let anything down through between the cells. The membranes are fused at the top, so all of the receptors are up here at the pore. Once this cell generates a generator potential, it's transferred to the afferent axon, or the process, of 7, 9, or 10, and that's how it becomes now an action potential and takes it to the brain stem. Okay? All right, going back to vision. So, your visual fields, retinal fields, nasal or temporal. From anatomy, you know that the temporal retinal axons project ipsilateral. The nasal retina crosses in the chiasm. Where they project is to a structure called the lateral geniculate body of the thalamus. The lateral geniculate body, if you look at it, is actually, so if that's the lateral geniculate, you got a bunch of cells here, you got a bunch of cells here, got a bunch of neurons here. In other words, it's laminated. Ipsilateral projections go into certain lamina. Contralateral projections go into the other lamina. And from there, the projections go out through the optic radiations to the primary visual cortex. These are the optic radiations here, coming out of the lateral geniculate. The point is that this lateral geniculate sees that visual field. This one sees that visual field. One thing I forgot to add on the hearing. No, we haven't done hearing yet. Oh, sorry. Brain farted there. All right. There's a guy named Brodmann, B-R-O-D-M-A-N, that in like 4000 BC, he looked at the cerebral cortex. It wasn't 4000 BC. You should have just said, that's stupid. No, it wasn't 4000 BC. He looked microscopically at the cortex and assigned numbers to cortex that looked the same. There are like 40-something different numbers that he assigned the cerebral cortex. Interestingly, as it turns out, decades later, the numbers represented similar functions. Area 17, Brodmann's area 17, is your primary visual cortex. Brodmann's area 18 and 19 are the association cortex. The primary visual cortex is right here in the occipital lobe on either side of that structure called the calcarine fissure, or sulcus. The gyrus that's on either side of that sulcus is area 17. 18 and 19 are the areas above and below the primary. Now, this illustration here puts a bunch more lesions into the visual pathway. Quickly, if you lesion the optic nerve, you're blind in that eye. Right? You lesion the optic chiasm, you have bitemporal hemianopsia. Because you get the crossing nasal fibers which will see the two temporal fields. You lesion the tract, either 4 or the entire optic radiations, 7, it produces the same thing. It's going to be a right or left homonymous hemianopsia. However, the visual pathway is more specific than that. The optic radiations see certain quadrants. The crossing fibers, like 3 here, I mean not the crossing fibers, the ipsilateral fibers, like number 3 here, if you get number 3, you see you're only going to lose that one nasal visual field. So you need to spend a little time with this. All right. A couple of relay type things we want to talk about. In addition to going to the lateral geniculate, that's the primary point for the visual pathway to the lateral geniculate body. You also have projections down into the brainstem, into the cranial nerve nuclei for cranial nerves 3, 4, and 6. 3 and 4, as I said, were in the mesencephalon. 6 is in the pons, as I said earlier. This way, you can track light with your eyes. If I'm going to look at something, I have to be able to rotate my eyeballs accordingly. So there's a very tight connection between the optic, the optic system and cranial nerves 3, 4, 6. Interestingly, also, there's a connection. So when we're talking about cranial nerve 3, there are 2 components of 3, right? There's a component that goes to the extraocular eye movement muscles, all others 3. So not the superior oblique and not the lateral rectus, but all of the other ones. And then there's a parasympathetic component of 3. What does the parasympathetic component of 3 do? It constricts your pupil. What dilates it? The sympathetics. The parasympathetics constrict your pupil. And there's a relay in your brainstem that allows you to do that based on the amount of light coming in. There has to be, right? So here's the optic pathway here, going to the lateral geniculate nucleus of the thalamus. There's your optic radiation. There's also a collaterals that go into an area of the mesencephalon, because that's where cranial nerve 3 is, that it has the parasympathetics, right? It's called the pretectal area. There's a synapse and then a distribution to both parasympathetic nuclei. The parasympathetic nucleus of 3 is called the Edinger-Westphal nucleus. There's one on each side of the mesencephalon. Edinger-Westphal. You okay? Somebody said something? All right, so if we go back, let me go way back. There's the motor nucleus of 3 that goes to the extraocular eye movement muscles. There's the Edinger-Westphal nucleus sitting right beside it in the mesencephalon. Okay. Here's the cool thing here about this nucleus. because it's bilateral, if I shine a light in this eye, what's gonna happen? It's gonna go down through here, it's gonna go down through there, it's gonna relate to that, that's gonna go out, it's gonna find its postganglionic in the ciliary ganglion, it's gonna go to the constrictor pupillae, and my pupil will constrict. That's the direct light reflex. Also, if I shine a light in this eye, it comes down through here, it synapses in the pretectal region, crosses to the other side, to the other Edinger-Westphal, out to the ciliary, and it will cause my other eye to constrict. That's called the direct and consensual light reflex. The direct and consensual light reflex. Have you learned that yet? That's fine, this is the pathway. Did you know this pathway already?
No.
Here's something that'll jack you up. I'm going to cut that optic nerve. I'm going to cut it. I am blind in my left eye. I shine the light in my right eye. This pathway is still intact. My left pupil will still constrict, even though I'm blind in that eye. If you shine the light in the left eye, neither pupil will constrict, because you can't get the light back to the Edinger-Westphal nucleus. Okay?
Are these nucleus like considered bilateral?
The input,
The
the input is bilateral. Let me tell you this. Most of the cranial nerve nuclei that we showed you in that schematic there receive a bilateral innervation from above. So when you have a stroke does not affect vision, your ability to work your tongue, muscles of mastication, hearing, balance, because it's bilateral. The only thing that a stroke does in your head and neck in terms of cranial nerves that you can outwardly see is the muscles of facial expression below your eye. This is the only place, well, actually there's another place, but right here is the most significant place where there's only a unilateral innervation from the cranial, from the descending input to the cranial nerves. If you stroke this side of your brain, not to worry. The other side can drive your tongue. That's why people with strokes never have a problem with swallowing. 9 and 10 and 12 and 5 all relate to swallowing. They're not impacted by a stroke on one side because the other side can take care of it. You can tell your mama that this weekend to learn something in the music. All right, hearing. The auditory pathway is really interesting because it is so it's so complex in terms of the number of relays, or the number of connections along the pathway. Here's your cochlea right here. You guys talked about the cochlea and the spiral ganglion. These are bipolar cells, right? I said cranial nerve 8 sits on the junction between the pons and the medulla. There are two nuclei that these spiral ganglion cells project to. They're called the dorsal and ventral cochlear nucleus. From there, projections go north through a thing called the lateral, the lateral lemniscus. You don't need to worry about that. They also have projections to the other side. So by the time you get to the top up here, hearing is bilateral. That's why you never lose hearing in a stroke, ever. The primary auditory cortex is out in the temporal lobe, right out here, all the way out here, supplied by the middle cerebral artery. You can just destroy one side, not to worry, the other side takes care of both ears. You never lose hearing with a stroke. There are stops in the brainstem with hearing. I'll talk about it in just a second. But appreciate that. There are stops in the brainstem. As a matter of fact, this is the dorsal side, right? The dorsal side. Let me go back to this thing right here. Here's the dorsal side, right? There are two bumps here. These are called the inferior colliculi. There are two bumps there. Those are called the superior colliculi on the back side of the brainstem. The inferior colliculi are relays for hearing. The superior colliculi are relays for vision. This area of the brainstem is called the tectum. This will come together here in just a second. So here's the inferior colliculus right there. From the inferior colliculus, hearing goes up to the medial geniculate body of the thalamus. The lateral geniculate body covers vision, the medial covers hearing. And from there, it's relayed to the cortex. There is a tonotopic relationship here that's interesting. If you take the snail shell of your cochlea, low frequencies are heard or detected best at the top. High frequencies at the bottom of the spiral. The axons, the input into these different nuclei and pathways is reflected onto the cortex, where high tones are detected in one part of the cortex, low tones in another. It's a tonotopic relationship. Very precise. This is what it looks like if you kind of draw it out together. You get a relay from the ears in through the brainstem, up to the thalamus, and then on over to the auditory cortex. I thought that was just kind of a cool slide. Doesn't add anything to the discussion, it's just a cool slide. Here's the cool thing, though, about hearing. I said that the relay went to the inferior colliculus, right? And that the inferior colliculus is part of the tectum, called the tectum. There is a tract that goes down into the spinal cord. It's called the tectospinal tract. You see right here, tectospinal tract. So I'm sitting here talking to Alex, and we're talking about this weekend, and all of a sudden, I hear the rattle of that rattlesnake. Meredith pulls the rattlesnake out of her purse. I hear it. What do? I turn my head in the direction of the sound. I don't turn my whole body in the direction of the sound, just my head, because the tectospinal tract only goes to the cervical spinal cord. It turns my neck, or turns my head in the direction of sound reflexively. You know, I'm walking along and Larry goes, and I turn my head. I hear it, I turn my head. The other thing is sight tectospinal. All of a sudden, I see a flash, I turn my head to the flash. So it's a relay of your cervical musculature that rotates your head in relation to sight, sound. In addition, you gotta rotate your eyeballs with it, right? So there must be a connection between the colliculi and the extra-eye loop muscles. And there is. So that as I rotate my head, my eyes fixate on Larry's flatulence. Yep, there it goes again. Any questions? That's kind of cool, huh? Same thing happens with balance. Balance is more complicated. Balance actually has 4 nuclei. Hearing has 2 nuclei, dorsal and ventral cochlear. Balance has 4. The vestibular apparatus has 4 nuclei on the pontomedullary junction that controls balance. It's connected with the cranial nerve nuclei for 3, 4, and 3, 4, and 6. And as I think I mentioned in anatomy, that is, the vestibular apparatus does 2 things. One, it detects acceleration, deceleration. Second, it detects angular motion, right? They've talked about that the past 2 weeks, right? There are little rocks inside your, okay, so we know all that. But in order to keep my eyes focused on the target, regardless of where my head is in space, that requires a connection between the vestibular apparatus and 3, 4, and 6. That's what this is right here. That pathway is called the MLF, the medial longitudinal fasciculus, if you're on Jeopardy. I don't care that you know that, just that you know there's a connection between the vestibular apparatus and 3, 4, and 6. Here's it kind of drawn out. Here's the vestibular nucleus here, input from the hair cells. You have different output though. One is through the musculature, so that you're able to manage your musculature. I mean, how is it that I can take my head and I'm not going to pat Annie on top of the head? I can do it regardless of where my head is in space. There has to be a coordination between that and my output. Otherwise, you couldn't walk. Anybody ever had vertigo? Vertigo? Meniere's disease, where there's an inflammation of the vestibular portion of this nerve. I mean, these people, they can't walk. It's horrible. You've got reflexes, as we talked about here, to the 3, 4, 6 are over here, and to the cerebellum, because certainly your vestibular nerve has to connect to cerebellum. What does the cerebellum do? Cerebellum modulates movement in motion. The cerebellum modulates motion, essentially. So that as I walk smoothly. I'm not all over the place here. I walk smoothly. That's the cerebellum doing it. If I'm sitting still, I still have a ton of muscle contraction going on. I'm just not moving. The basal ganglia manages that, modulates that. It's the cerebellum that modulates muscle function when you're in motion. So when you're doing the physical exam, you want to test both of those. Their ability to sit still and their ability to do things in motion. The vestibular nerve is part of that. Okay. That's all I know about this. Any questions? Yes, ma'am.
For the nose, as far as the location of the cilia, is it all throughout, like the turbinates and everywhere in there, or is there just one area where they're?
No, it's just at the very very top of the nasal cavity, all the way up here. There are no olfactory receptors anywhere else in the nose except at the very top, above the superior concha. Above the superior concha. Very small area. Anything else? All right, well, enjoyed seeing you guys again. See ya.
Bye.
Bye.